Imagine a world where cells don't just react to their environment, they communicate with it, like a language written in proteins, signals, and timing. What if we could decode that language? Not metaphorically, but mathematically, computationally, and predictively. Today, we're diving into a groundbreaking new framework called Hierarchical Molecular Language Models, or HMLMs, a fusion of artificial intelligence and systems biology that redefines how we understand cellular signaling. This isn't just another neural network. It's a molecular language model, trained not on words, but on the very grammar of life. Molecules as tokens, interactions as syntax, functions as semantics, and coordinated cellular responses as narrative. Let's break it down, from first principles to real-world impact. Hash the problem, why traditional models fall short. For decades, scientists have modeled cellular signaling using tools like ordinary differential equations, ODEs, Boolean networks, and Bayesian models. But these approaches hit walls. ODEs are precise, but require exhaustive parameterization. They fail at scale. Boolean networks simplify everything to on or off, losing critical nuance. Bayesian models capture uncertainty, but struggle with feedback loops and dynamic crosstalk. And here's the core issue. Biological systems aren't linear. They're hierarchical, context-dependent, and temporally rich. A signal from TGF-beta doesn't act in isolation. It interacts with mechanical strain, metabolic state, and other pathways, all in real time. Traditional models can't capture this complexity, especially when data is sparse, noisy, or incomplete. Hash the solution. HMLMs, a molecular language model. Enter HMLMs, a novel architecture built on the transformer foundation of large language models, but adapted for biology. Here's the core insight. Cellular signaling is a language. Tokens are molecules, receptors, kinases, transcription factors, even protein complexes. Syntax is defined by physical and chemical rules, who binds to whom, when, and how. Semantics are functional outcomes, activation, inhibition, translocation. Discourse is the coordinated activity of pathways, the story of a cell's decision: grow, migrate, die, or fibrose. Just as transformers learn to predict the next word in a sentence, HMLMs learn to predict the next state of a signaling network, but across multiple scales. And they do it using graph-structured attention, not sequential processing. Hash how it works, from molecules to meaning. At its heart, an HMLM treats every signaling molecule as an information transducer, a system that receives inputs, processes them internally, and produces outputs. Formally, this is a tuple. Input space, output space, state transition function, output function. Input space, external signals, ligand concentrations, ATP levels, stress cues. State space, internal configuration, conformational states, activation status, phosphorylation, localization. Output space, downstream signals, phosphorylated substrates, gene expression changes. These transducers aren't isolated. They're connected in a directed graph, the signaling network, where edges represent information flow. Now comes the key innovation: hierarchical multiscale attention. HMLMs don't just process the network at one level. They operate simultaneously across molecular scale, individual proteins and their states. Pathway scale, functional modules like MAPK, Pi3K, row signaling. Cellular scale, integrated responses, growth, apoptosis, migration. This hierarchy is enforced through scale-bridging operators. Aggregation, combining molecular activity into pathway-level signals. Decomposition, distributing pathway-level outputs to individual components. Translation, converting data formats across scales. It's like having a translator between micro and macro, ensuring that what happens at the protein level informs the cell's behavior, and vice versa. Hash architecture, the engine of prediction. The HMLM architecture is a multi-layered system. 1. Multimodal input embedding. Phosphoproteomic, transcriptomic, imaging, and perturbation data are fused into a unified representation. 2. Graph-structured attention. Unlike standard transformers, attention is computed only over neighboring nodes, respecting the network's topology. 3. Temporal dynamics layer. Each node gets a time-dependent embedding, capturing the timing of activation, decay, and feedback. 4. Hierarchical attention. Within scale and cross-scale attention, mechanisms allow the model to learn both local interactions and global coordination. 5. Ensemble prediction heads. 3 specialized regressors, molecular, pathway, and cellular, each trained to predict different aspects of the response. And crucially, attention weights are learned, not assumed. The model starts with prior knowledge, network topology from curated databases. Then, during training, it refines those connections using temporal correlation analysis across multiple time lags. This hybrid approach, structure plus data, gives HMLMs both biological interpretability and predictive power. Hash results, performance that outshines the competition. We tested HMLMs on a complex cardiac fibroblast signaling network, over 100 molecular species, 11 functional modules, 200+ regulatory edges. The results: a 30% improvement over graph neural networks, GNNs, and a 52% improvement over ODEs. Mean squared error, MSE. HMLM achieved 0.058 compared to GNN's 0.083 and ODE's 0.121. Under sparse sampling, With only 4 time points, HMLM maintained an MSE of 0.041, while others degraded dramatically. Why? Because HMLMs don't just fit data, they infer missing states. They use the network structure and temporal patterns to fill in the gaps. And the predictions were biologically meaningful. TGF-beta right arrow SMAD3 activation. Attention weight of 0.72, confirming a canonical profibrotic axis. SMAD3 right arrow YAP, 0.61, revealing a previously underappreciated crosstalk between TGF-beta and mechanotransduction. PDGF MAPK, 0.76, validating a known pathway. Mechanotransduction right arrow fibrosis, 0.69, showing how mechanical cues drive disease. These aren't just numbers, they're discoveries. # biological insights, beyond prediction. HMLMs aren't just accurate, they're insightful. Through attention analysis, we discovered previously unknown crosstalk between mTOR and STAT3 pathways, later validated experimentally. Dynamic shifts in pathway dominance. Early receptor signaling gives way to feedback-regulated responses. Compensatory activation patterns after inhibition, explaining therapeutic resistance. Convergent regulation of fibrosis markers from multiple pathways, suggesting multi-target therapies may be more effective than single-pathway drugs. This is the power of AI-driven biology, not just prediction but hypothesis generation. # why this matters, the future of precision medicine. HMLMs are more than a model, they're a paradigm shift. They bridge molecular mechanisms with cellular phenotypes. They scale predictably. They integrate multi-omics data. They work under real-world constraints, sparse, noisy data. And they're interactive. You can simulate drug perturbations, predict resistance, and design combination therapies, all in silico. In precision medicine, this means personalized signaling network models for individual patients, predictive diagnostics based on pathway-level interpretations, adaptive treatment protocols that evolve with patient response. Imagine a future where a clinician doesn't just prescribe a drug, they simulate its effect on a patient's unique signaling network and choose the best combination up front. That future is closer than you think. # challenges and future directions. Of course, no model is perfect. We rely on curated networks, which can bias results toward well-studied pathways. Future work will explore structure learning, letting the model discover connections from data while respecting biological constraints. We also need validation on experimental time series data, phosphoprotomic, transcriptomic, and imaging datasets to confirm that learned attention patterns reflect real biology. And we're just scratching the surface. Integrating single-cell data, spatial context, and even foundation models pre-trained on vast biological datasets could unlock deeper insights. # conclusion, a new language for life. HMLMs represent the convergence of two worlds, artificial intelligence and systems biology. By treating cellular signaling as a language, we've built a model that doesn't just simulate, it understands. It learns the grammar of life, the syntax of interaction, and the meaning behind the signals. This isn't just about better predictions. It's about deeper understanding of how cells make decisions, how diseases emerge, and how we can intervene. As we move into an era of AI-enhanced biological discovery, frameworks like HMLMs won't just be tools. They'll be co-investigators, helping us ask better questions, test bold hypotheses, and accelerate the path from lab to clinic. The language of life is complex, but now we have a way to speak it. All code and data are publicly available at https://github.com/HassieHayes/HMLM. For more on this research, contact Hassie at UARC, Edu. This is the future of biology, not just data but meaning. Not just models but insight. Not just prediction but understanding. Stay curious, stay critical, and keep pushing the boundaries.